gentlemen. Thank you for opportunity to present our research focused on utilization of Hitchcock protein in induction of Th1 and CD8 T cell response toward P24 antigen derived from HIV virus. I'm from Olomouc, which is a university in Czech Republic, which is second oldest one and rapidly growing. Now we are having 3 buildings allowing us to do not only teaching but also research. And what is our research focused on? Concerning to vaccine-induced protection, it's well known that the majority of vaccines protect through, or majority of known clinically approved vaccines protect through antigen-specific humoral immunity. And this humoral immunity has limited effects against many viruses and tumors, and were paralleled with activation of antigen-specific humoral and cell-mediated immunity, which is very important and needs to be induced. There are several approaches how cell-mediated immunity could be induced, of which, for example, various liposomal formulations or various clinically tested formulations are available. In activation of CD8 T cells, there is one interesting point which is at present time known, but I think it's good to mention it here, that antigen-specific immunity is definitely initiated by antigen-presenting cells, and antigen-presenting cells are involved in activation of both CD4 and CD8 T cells, but through presentation of peptides on different MHC molecules. And concerning to presentation for CD8 T cells, MHC class 1 is important. But in general, MHC class 1 is presenting only endogenously produced antigen, and furthermore, majority of antigen-presenting cells, especially dendritic cells, are not those which are affected by, for example, infection or tumor. So they should present during activation of naive CD8 T cells antigen which they encounter from then surround. Exception are, for example, DNA vaccines which are targeted to dendritic cells, but in general, presentation of exogenously encountered antigens needs to be performed by dendritic cells on MHC class 1. Mechanisms involving this phenomenon are termed as a cross-presentation, and there are several pathways which were described to be involved in cross-presentation antigen. In all of those pathways, with exception of gap junction, cross-presentation endosome, early endosome is the first organelle which is encountering antigen, exogenous antigens, which is further either released into cytosol to be processed like, for example, viral antigen in proteasome and present on MHC class 1, or are released to cytosol but transported back to early endosome and processed by replacement of peptides on MHC class 1 molecules which are continuously recycling from cell surface. In some cases, it was shown that also exogenous antigens could be directly processed by cathepsin S and presented on recycling MHC class 1 molecules. Hitchcock proteins are unique group of proteins which are involved in processing of nascent proteins, but in 1984 it was shown, and following several reports confirmed, that the Hitchcock proteins could be used for immunization, protecting from tumor development. It was shown that preparation of Hitchcock proteins, mostly using ADP column from tumor cells, could induce cytotoxic T cells against the same type of tumor. And later it was shown that not the Hitchcock proteins on their own, but associated peptides of small molecular weights are the target of antigen-specific immune response. This observation was modified by experiments which showed that also covalently linked proteins or peptides to Hitchcock protein could be also transported and could be target of antigen-specific cell-mediated immunity. Concerning to Hitchcock protein, several mechanisms were described to be responsible for processing and presentation of exogenous antigen on MHC class 1. And they include both that transport to cytosol, or sometimes called as a cytocellular pathway, where proteasome is important, especially if C-terminal fragments of that associated protein needs to be presented on MHC class 1. Or they could also utilize that vacuolar pathway, which is relying on processing of peptides or protein associated with Hitchcock proteins just in early endosome and association with recycling MHC class 1 molecules. Unique property of Hitchcock proteins were described especially for Hitchcock protein 70, Hitchcock protein 90, glycoprotein 96, and to some extent even to Hitchcock protein Hitchcock protein 90. And it was shown that they are recognized by several receptors which are important for endocytosis of Hitchcock protein with associated antigen. Other receptors were shown to be responsible for activation of dendritic cells, and this is a very important aspect of Hitchcock proteins, that they could serve as an endogenous danger signal, and they could contribute to activation of antigen-presenting cells. Because not only transport of peptides to endosome is important, but also activation of dendritic cell is the crucial for their ability to stimulate naive CD8 and CD4 T cells. In our experiment, we were focused, as I mentioned, on HIV P24 antigen, which is one of promising antigens used for elicitation of cell-mediated immunity, which could contribute to protection, especially during set point of viremia, which occurs within 3 to 6 months after transmission of HIV virus. And they could contribute to setting lower viremia during set point, and especially GEC protein and POL proteins are those targets recognized by CD8 T cells and partially CD4 T helper 1 cells. Concerning to those proteins, the main advantage of those proteins is their quite a higher stability or conservancy in comparison to envelope protein, which is frequently used as a target for elicitation of neutralizing antibodies, which face the main problem with ENV variability. P24, on the other hand, is relatively poorly recognized by dendritic cells. Therefore, some aspects which could facilitate transport of P24 for dendritic cells and for their engulfment will be very helpful, which is one of property of HSP protein. And P24 targeting to dendritic cells is therefore very important. We prepared several constructs based on murine, HSP17, 70, and P24, and those constructs differ in orientation of the recombinant HSP70 and P24. They were produced as a fusion protein expressed in E. coli, and for comparison we use just HSP70 and P24 alone. Those proteins were purified after expression in E. coli using nickel nitryl triacetic acid column affinity chromatography, followed by phase extraction of lipopolysaccharide using Triton X114. And this extraction was performed until the concentration of lipopolysaccharide was below 2.5 unit per milligram of protein, which is acceptable as a concentration of lipopolysaccharide, which is not inducing lipopolysaccharide specific inflammation or lipopolysaccharide specific activation of dendritic cells in vitro. This is just gel showing individual recombinant proteins, which were confirmed by mass spectrometry just to confirm their identity. One, I think, important fact is that native purification procedure we used for purification of those proteins was effective in keeping ATPase activity of individual HSP preparation, as is shown here, because in some reports it was shown that HSP70 protein as such, if is not contaminated by lipopolysaccharide, is unable to stimulate dendritic cells. But in those reports, they didn't confirm their, let's say, ATPase activity, which could be some indicator of natural conformation of all those proteins. We use those preparations for in vitro experiments trying to demonstrate activation of bone marrow derived dendritic cells after 24-hour exposition of those bone marrow derived dendritic cells to individual antigens, and we analyzed expression of CD40, CD86 as activation markers on dendritic cells and MHC class I molecules. And it was demonstrated that especially HSP70-P24 fusion proteins was highly effective in stimulation of costimulatory molecule CD86, which is even higher than control lipopolysaccharide stimulation, and was much higher than just used medium. This is just a confirmation of lipopolysaccharide dosage calculated for each individual proteins. This dosage was calculated from Limulus amoebocyte lysate assay, showing that this is not effect of lipopolysaccharide. The control lipopolysaccharide contained 100 nanograms per ml in vitro. Further, we analyzed whether these fusion proteins could somehow facilitate endocytosis of associated P24 antigen by confocal microscopy, in which we try to colocalize P24 using Fitz-labeled antibody against P24 and early endosome using early endosome specific antibody labeled with red color. And we found that analysis of several cells, this is just an example, confirmed that HSP70 P24 fusion protein is the best one which is engulfed by dendritic cells by analysis of Pearson colocalization coefficient. Further, we approached the experimental immunization of mice using individual recombinant protein. As I mentioned, Hitchcock protein as such are able to stimulate dendritic cells, and therefore for such experiment we didn't facilitate the immune response by co-administration of any adjuvants to analyze just the pure response against the protein. So we immunized 5 mice per group by individual protein, either P24 fused with HSP70, HSP70 fused with P24, and this is just mixture of two recombinant proteins, not fusion proteins, P24 and HSP70 and control. Dosage of the protein you can see are different, but the dosage was calculated to be equal on molecular concentration, not on weight concentration of the protein. And immunization was performed twice, priming and following after 40 days by booster. We analyzed also humoral immune response, because for Hitchcock protein it was shown that the humoral response could be elicited by Hitchcock protein fused antigen, and we analyzed immunoglobulin G in isotype IgG1, IgG2A, IgG2B. Those two are representing mostly T helper 1 type of immunoglobulin in mice, and IgG1 is T helper 2 type immunoglobulin in mice, and this is total level of immunoglobulins. Here we can see that immunization of mice with HSP70 P24 or P24 HSP70 is able to stimulate substantially higher increase in serum concentration of P24 specific antibodies to contrary to P24 alone or just mixture of P24. Similar observation was made for IgG2B, which is here shown to be statistically significant. We observed a higher level or increase in P24 specific antibody as a total immunoglobulin, even for P24, but still the differences are quite high. Further, we analyze cell-mediated immunity by ELISpot analysis on interferon gamma secreting cells from mice splenocytes after stimulation for 4 days with depyrogenated P24, and we show that HSP70 P24 fusion protein is the best one for stimulation of murine T cells, which were later producing in vitro interferon gamma after re-stimulation. In summary, we prepared fusion proteins and HSP70 and P24 protein with low concentration of endotoxin, which is accepted to be applicable for immunization experiment and is accepted to be not stimulated immune response by contaminating lipopolysaccharide. We show that P24 HSP70 elicit P24 specific antibodies better than non-fusion protein or P24 alone, and especially HSP70 to P24 in this particular orientation elicit most P24 specific interferon gamma producing splenocytes. HSP70 P24 is also effective in driving of P24 to early endosome, and HSP70 fusion proteins in general, both orientation, increase expression of activation marker in pulsed bone marrow dendritic cells. On the end, I would like to acknowledge researchers from our laboratory and collaborators from France, which provide us with P24 sequence and statistical analysis performed by colleagues from University of Alabama at Birmingham. Thank you for your attention.
What's the fusion between the P24 and the HSP? Have you got a linker, or is it?
It was fusion protein based on preparing of recombinant plasmid coding for those proteins. So this was just sequence of P24 followed by HSP70, and in another plasmid HSP70 sequence followed by P24. So it was expressed as a fusion protein as such.
I understand that. But so did you synthesize the DNA as one protein, or did you actually, how did you fuse
We isolate the DNA4, HSP70
Yeah,
from murine cells.
right.
Use that DNA for P24 and just clone it together in one expression plasmid, and in another we clone it in opposite
Okay.
orientation.
But there's no linker, just straight sequences straight
It
together?
was tried.
Okay.
It depends, the linkers depends on expression. We tried some linkers,
Yeah.
but this trice connection was among those best, so we didn't try to introduce any further sequencing.
Okay, fair enough.
So in your interferon gamma spot assay, do you have any idea whether those interferon gamma producing cells are CD4 or CD8?
So we didn't analyze yet those experiments, but they are ongoing, so we will know soon. But for ELISpot, it is not for us as easy, so we decide to analyze it by intracellular production of interferon gamma through flow cytometry.
And I might have missed it, but in your ELISpot assay, what antigen did you use to stimulate?
We use recombinant P24 without any fusion partner as a text for purification, and this P24 was LPS free, or with a concentration below that 2.5 units per milligram.
Thank you.
So we didn't stimulate with peptides.
Where this Hitchcock protein is normally expressed with the single lines of autoimmunity induced by your vaccine?
Whether they induced autoimmune responses?
Yes.
It's still open question, but in majority of reports used HSP for immunization, there were not observed mark of autoimmunity because they could elicit some cross-reaction if, for example, Mycobacterium HSP is used for immunization. But for those concerns, we prefer to use murine HSP70 as an autologous antigen. But they should be generally well tolerated due to their function because they are still released out of dying cells, and the autoimmunity is not observed to be directly linked to HSP protein. Thank you.